Right. So thanks very much, Bill. So we're at time. So we'll as previously discussed, if you want to capture any questions for Bill in the app, we'll pick them up later. So next, I'd like to introduce and welcome Jenny. So Jenny Wei, who's head of R&D Informatics and Technology at Kite Pharma. Over 23 years of experience in the pharmaceutical industry, delivering innovative digital solutions, advanced analytics for small molecules, biologics, and cell therapy. The title of Jenny's talk is Building Cell Therapy Multimodal Scientific Ecosystem.
Thanks, Dave. Hi, everyone. My name is Jenny Wei. I'm from...
Steer the microphone.
Oh, sorry.
Thank you. I appreciate it.
My name is Jenny Wei. I'm from Kite, a Gilead company. So my background is in R&D informatics, and I have been working for biopharma R&D in the last 24 years. Today, I will co-present our experience in building cell therapy multimodal research ecosystem with Andres from Deloitte. So I will let Andres introduce himself before we jump into the presentation.
Thank you, Jenny. Good afternoon, everybody. Thank you for being here, especially those of you who are running enthusiasts. I know that today we are competing with the Boston Marathon. Tall order. I can promise you this will be almost as exciting as the race was. My name is András Fáncsik. I'm a leader in Deloitte Consulting's Life Sciences division, focusing on R&D, data management, and data analytics solutions. Coming from a medical and scientific background, focusing on cell and molecular biology and neuroscience. And I'm very, very excited to be here with you today and have a chance to talk to you a little bit about the multimodal data platform solution that Deloitte had the opportunity to assist Kite Pharma with. Jenny, back to you.
Thanks. A few disclosures. The work is funded by Kite Pharma. The view expressed here are from our speakers, not from the company, not from the company. The conference sponsors did not really influence the contents of this presentation. With that, I'm going to kick off as short introduction to Kite Pharma. Kite Pharma is a global cell therapy leader. The company started in 2009, and it was acquired by Gilead in 2017. So our company has two products approved with 5 indications. The products are marketed in more than 25 countries. Since the first approval in 2017, we have treated more than 19,500 cancer patients. As of today, Kite Pharma has more than 4,000 employees and more than 1 million square feet of manufacturing and R&D lab space. So what Kite has developed is autologous cell therapy. So this is a game changer for cancer treatment, and it has the potential to cure the cancer. In the recent years, the cell therapy has advanced beyond cancer treatment to fight autoimmune disease. So Kite has industry-leading research and also manufacturing capabilities. So Kite has a mechanism putting in so we can ensure the 14-day, day-to-day, vein-to-vein turnaround time for the treatment. So in the overall discovery process is a complicated process. So during this process, many different data modalities are created from different business capability groups. So first, we have a big flow cytometry core. Flow represents more than 60% of active data of the whole cell therapy research. This technology has been widely used for cytotoxicity, cell proliferation, sorting, immunophenotyping assays. Genomics, single cell, RNA-seq, predominantly today, ATAC-seq, CyTOF, These are very crucial for us to understanding disease relapse and recurrence mechanisms. Pathology, traditional immunohistochemistry, and the most recent single-cell spatial multi-omics. These are very crucial for cell therapy to get into a solid tumor. Last, synthetic biology, right? So in the whole CAR discovery, genetic engineering is a very important factor. So these technology help to enhance and to engineering the CAR. So each data modality has a set of tools for data acquisition and data analysis. Largely, these tools are siloed. So on top of it, we have a LIMS platform or electronic lab notebook platform. So in building such a research ecosystem, we try to use the LIMS platform to connect these different systems together, make them talk in the cloud. So here comes our design principle and our thinking behind the ecosystem. So our design principle is data fairness. So our 3 pillars, cloud native, building cloud native and the digital LIMS platform as a foundation. Second, automation focused. Everything we want to make digital flows, scientific flows, data pipelines, even data, metadata, all these be automated, get to connected with the LIMS. Third pillar is what we call prediction capable. With machine learning data, how we can augment the CAR discovery. So our digital journey has been going on and has 3 phases. So we have come a long way to build foundation in phase 1. And we are right now focusing on the automation phase to mainly try to reduce the manual data entry to reduce user burden. And we are starting to pilot into the prediction capable phase. So I'm not going to go to details on the slides on the challenges. These are, I think, common in pharma, like from data intake to data storage and analysis, sharing, and also the making silo the data integrated. All these are common challenges. So use flow as an example to demonstrate these challenges, right? File naming convention, inconsistent, and move the file, from network file system to laptop to do the analysis. Flow is still the traditional method, right? It's just computationally limited, and a lot of time wasted moving data over to the laptop. And there's no, obviously, robust search capability. A lot of problems, you see duplicated, triplicated FCS files everywhere. And also the other challenge is still, using LIMS, but data entry is very manual, cutting paste into a table, with a schema. All these challenges, scientists has to face it day to day. So how to tackle this? Like phase 1, which achieved the new real-time transfer of raw data from instruments directly to cloud. We set up cloud computing and the primary analysis pipelines. And we built inventory management biomaterial registry in the LIMS and the ERN. And we also have a good scalable cloud storage for raw and processed data. So this is just a overview of what different technologies we used for getting cloud native for different data modalities. So using flow cytometry as an example, I will have Andres to talk about details.
Thank you, Jenny. So as Jenny mentioned, basically the 3 pillars that we were pursuing: cloud native, automation enabled, predictive capable. Let me tell you a little bit more about the specifics that we were trying to accomplish when we pursued cloud native. The specific example that we're showing here is the flow cytometry related modality that we built out. And our immediate objective was to take advantage of the cloud, enable the organization to utilize all the benefits that a cloud native platform would give you, whether you're thinking about scalability, high performance computing, the ability to promote centralized governance, ability to easily scale storage, potentially create a standardization advantages. All of this is what comes from getting data as fast to the cloud as fast you can. So as you can see on the left side, what we started with is created a mechanism that very quickly grabs the native assay files off of the flow cytometer instrument and immediately moves it into the cloud. The software that we chose for this is the provided by the Magito, which was already present at Kite's environment, and it does a great job with grabbing files and putting them into AWS S3 buckets. And from there, we were able to take advantage of the AWS Cloud Compute services to process these files, interact with the metadata, and move them around, standardize them, etc. And ultimately, through API interfaces and an API gateway by Apigee, we were able to automatically register these data files. And in the case of flow cytometry, as you are probably familiar, single experiment creates hundreds of files. So that was definitely advantageous to immediately being able to automatically create registration of all of these files in the ELN system, which in Kite's case was Benchling. And from there, the files were moved into a Kite data repository, which was also supported in the cloud by a high-performance, highly extensible FSx ONTAP file system that AWS provides. We created a metadata repository where everything was automatically registered in the DynamoDB. And then finally, we were able to actually help with a cloud-based implementation of Flojo on AWS AppStream. So that completed the cloud native circle and allowed us to accomplish this major target, major goal. Back to you, Jenny.
Thanks. So in phase 2, we are concentrating on the automation part. So what we achieved is we have a batch file transfer with event trigger mechanism. So we automate the EMA, the LEMS, entries. We have a way to getting entries with a link to raw data and the process data directly into LEMS. And we also enhance the analysis workflows, end-to-end to streamline it in the cloud. And also we help to bring raw data and also results back from the analysis workflow directly to the cloud storage instead of asking user to upload. So again, I will pass to Andres to talk about flow cytometry, how we do it.
Thank you. So how did this look like in the case of our flow cytometry example? So automation focused. Our main goal was basically to reduce any kind of manual work. And as we all know, the aforementioned copy and paste, that is kind of a bane of a scientist's existence. We definitely wanted to make sure that we have as minimal as possible. So in terms of the steps that I just described previously, every step we introduced automation based on triggers. What we started with is we allowed the researchers to review the native FCS files that were generated on the flow cytometer, And once they were satisfied that all the compensation, voltage, everything else shows up the way that they wanted, we allowed them to export into a designated folder. That folder was monitored, and the files as they showed up automatically were copied over to S3. So that basically was completely eliminating any kind of manual copy-paste exercise. As I mentioned, when this needs to happen by the hundreds, it's definitely advantageous. Once the files were processed, the API-based registration in Benchling was happening, where our automation was basically able to take advantage of the metadata extraction and metadata injection processes that FCS files allow. And so we were interacting with the metadata, we were interacting with the files, native metadata components that are text within the files, as well as the files, file name, and attribute-based metadata. So we were updating file names, we were updating storage locations, all of this happening based on the governance rules, business rules that were built into the system. So again, eliminating the end users, eliminating the need for the end users to interact with this process at all. And then finally, once the files were stored in their final location in the FSX ONTAP directory, we also built an extra step that basically allowed researchers to trigger a WSP file creation process. So Flojo being the primary analytics tool, we made it easier for researchers to select whatever files they wanted to analyze and do the gating process, and then auto-created a WSP starter file for them and allowed them to record all of custom attributes that they want to actually associate with those hundreds of individual samples, whether they were effector cells, target cells, plasmids, etc.
We skipped there.
Yeah, just a little bit about the overall architecture. So this picture shows the end-to-end technical architecture, which I'm not going to go through all of the numbers. Unfortunately, we don't have enough time, although it would be quite fun for those of you who are technically inclined. Oops. Can we jump back just a little bit? Enough to say that basically what I'm trying to emphasize here is that there are many steps, processes that are enabling that automation, all of them taking advantage of the cloud-native AWS software as a service solution. The other bookend components being Benchling, or on the left side, Limagito, playing very nicely with the cloud-based center. So it was really enabling us to build a very nice end-to-end cloud-focused solution. Back to you, Jenny.
Thanks. So I will quickly go through phase 3. It's a most exciting phase. So we are currently experimenting the data cataloging and exploration with the language model. So we try to reduce the manual data annotation and to allow natural language query to the data in a self-discovery to extracting the information from the data. So second topic, we are thinking about using machine learning, ready data, be able to augment the card discovery. Right, this is from three angles. Develop a card language model, try to predict the key properties of a bundle, right, the bundle interaction, and optimize the experiment design with historic data, and eventually targeting to card de novo design. And then lastly, very importantly, is image, right? Increase imaging on the space. We have pathology, spatial omics, radiomics, and also optic genomics as the latest. So the image this analysis with the language model is definitely an area worth to pursue. So I will get to and just talk about a little bit about what's the intelligent search.
And just very quickly, I'm realizing that we are running over time. So one thing that we wanted to enable on that prediction-capable journey is intelligent search. And what does that mean? Basically, we wanted to enable the users to provide a natural language question and find, in a large repository, find relevant files that are pertinent to that question. So for instance, somebody would ask, well, show me any experiments where we were focusing on CD19, CD20, or CD19-20 bicistronic targeting, right? And then the engine would immediately come back with a list of files that the users could further examine. what we did is basically created or used a Gen AI LLM model to create a vector embedding of all the data in the file repository, whether those were Microsoft Word docs, PowerPoints, Excel files, FCS files, WSP files, all of those can be in play. Create the vector representation, then created a vector representation of the query itself when it was happening real time, matched the similarity between the two, and the engine was force ranking based on relevance, force ranking the results, and in the UI, showing the users that these are the relevant files for you. So that we think is quite superior to the traditional way of doing it. It eliminates all of the tagging, annotation, and frees up really highly skilled subject matter experts to focus on what they want to focus is basically science.
Okay, so quickly talk about lesson. We can talk at the panel discussion time, but automation first. This is number one to us. You minimize data entry, manual data entry, reduce burden. This is very critical for the success, for the retention of the use of the LIMS. And lastly, this is, I see as a design pattern, right? Cloud orchestrated SaaS ecosystem. This is a way, to make all these different silo system talk. So lastly, I want to thank all the people made great contributions to this project and supported our digital journey from Kite Research to Kite R&D Information to Gilead IT, and also our collaborators from Deloitte and the AWS. Thank you all.
Thank you.